Hi everybody, in this video we are going to discuss about the life cycle of Plasmodium vivax. Life cycle of Plasmodium vivax. And we know that this Plasmodium vivax is a malarial parasite. So we are going to discuss about the malarial parasite, how it is completing its life cycle. So here what we are going to discuss is about the malarial parasite. So here if you are looking at this malarial parasite, we already said that it requires two hosts. Okay, we said that it requires two hosts. It requires two hosts. So that's why we will be saying that it is digenetic. Okay, it is digenetic. And we know that what do you mean by the word digenetic? Digenetic means what? When a parasite is using two hosts to complete its life cycle, we call it as digenetic. So here the Plasmodium vivax, the malarial parasite, and this Plasmodium vivax is what? A protozoan. It is a protozoan. It is coming under the protozoan, and this is causing what? Malaria, and it is requiring how many hosts? Two hosts. So that's why we are calling it as digenetic. So here in the how we are going to now discuss how this Plasmodium vivax will be completing its life cycle and how it is causing malaria in the human beings. How it is causing malaria in the human beings. Now if you are seeing this particular Plasmodium vivax, okay, requires two hosts. Okay, as we said here, it requires how many hosts? Two hosts. Okay, it requires two hosts, and the first host is what? The human being. Let us say human being. Okay, where it will be completing its asexual life cycle. So here we are saying that asexual life cycle. Okay, asexual life cycle will be taking place in the humans. Okay, asexual life cycle, or otherwise we can call it as what? Schizogony. Okay, we can call it as what? Schizogony. Okay, schizogony in man. Okay, this is taking place in what? In man. Okay, schizogony in man, we can say. So asexual life cycle, or otherwise we are calling it as what? Schizogony in man. Schizogony in man. And the other one is the sexual life cycle. Okay, sexual life cycle. Okay, sexual life cycle. Sexual life cycle. Otherwise, this is called as what? This is called as what? Sporogony. Okay, sporogony. Okay, sporogony in which organism it will be taking place? In female Anopheles mosquito. In female Anopheles mosquito. Female Anopheles mosquito. Okay, so here the life cycle of Plasmodium vivax is taking place in two organisms. In two organisms. That is what? One is in man and the other one is what? The female Anopheles mosquito. In two organisms it is completing its life cycle. Plasmodium vivax is completing its life cycle in two organisms, or two hosts we can say. One is in man and the other one is female Anopheles mosquito. So here in man it is completing its asexual life cycle, and in the female Anopheles mosquito it is completing its sexual life cycle, otherwise called as what? Sporogony in female Anopheles mosquito. Sporogony in female Anopheles mosquito. Now we are going to discuss about the asexual life cycle, or otherwise called as what? Schizogony in man. Asexual life cycle, that is called as what? Schizogony in man. Schizogony in man. So now we will discuss about the first part, that is asexual cycle. Asexual cycle, that is called as what? Schizogony in man. Okay, schizogony in man, which is here the word. If you are looking at the word schizogony, it is nothing but schizogony is a method of what? Asexual reproduction. It is a method of asexual reproduction. So here the Plasmodium vivax, okay, the Plasmodium vivax, they will be undergoing asexual reproduction in the human beings. In the human beings. So here, please remember, schizogony is a process of asexual reproduction in which the Plasmodium vivax undergoes asexual multiplication, or it is multiplying by asexual method. Okay, asexual method, or otherwise we can call it as asexual reproduction. So where this asexual reproduction is taking place? Asexual reproduction will be taking place in the man in cells called as what? The liver cells. It will be taking place in the liver cells, and as well as in the RBC cells. Okay, in the RBC cells. Okay, it will be taking place in the liver cells and also in the RBC cells. So you have to remember, the schizogony is what? It is a method of what? Asexual reproduction, by which what is multiplying? Plasmodium vivax is multiplying. Where this process is taking place, it is in the man or in the human beings. It is taking place in the human beings. So in the human beings, where do this process takes place? It is taking place in the liver cells and it is taking place in the RBC cells. So now, when it is taking place in the liver cells, we can call it as hepatic schizogony. Okay, hepatic schizogony. We call it as what? Hepatic schizogony. We call it as hepatic schizogony. And when it is taking place in the RBC cells, it will be called as what? Erythrocytic schizogony. Okay, erythrocytic schizogony. Erythrocytic schizogony. We call it as erythrocytic schizogony. Okay, this is what we have to remember, these two words now. So here in human beings, okay, Plasmodium vivax is undergoing what process? Asexual reproduction. What is the other word that we are using for the Plasmodium for asexual reproduction? It is called as schizogony. So we are using two terms here. If it is taking place in liver cells, we are calling it as what? Hepatic schizogony. If it is taking place in RBC cells, we are calling it as what? Erythrocytic schizogony. Okay, erythrocytic schizogony we are calling it as. Okay, these are the two terms which we have to remember. Now, how this process will be taking place? How this process will be taking place? Let us start like this. When an infected female Anopheles mosquito bites a healthy person. Okay, when a female Anopheles mosquito, infected female Anopheles mosquito means what? That mosquito is carrying the Plasmodium vivax. It is carrying the Plasmodium vivax at a particular life stage. Okay, this vivax, Plasmodium vivax, is present in the mosquito at a particular stage. And that stage will be released into the human body now. Now let us see this now. When an infected female Anopheles mosquito bites a healthy person, now that female Anopheles mosquito, okay, female Anopheles mosquito releases, bite, let me say like this, bite of female Anopheles mosquito will be releasing, the Plasmodium vivax, which is at an infective stage called as what? Sporozoid. Please remember, this word is very important one. Sporozoid. Sporozoid is one of the stage of the Plasmodium vivax. Sporozoid is one of the stage of the Plasmodium vivax. So this can be called as what now? It is the infective stage. Please remember that. Infective stage. So what is the infective stage of Plasmodium vivax means? Your answer should be sporozoid. What is the infective stage of Plasmodium vivax? Answer is sporozoid. So sporozoid is released into the body of a healthy person. So how it is released? By the bite of a female Anopheles mosquito. When the mosquito bites, along with its saliva, it is also releasing what? This sporozoid into the body of a healthy person. And this sporozoid now reaches to what? It reaches to the liver cells and also to the RBC cells. And once this reaches to the liver cells and RBC cells, which is at different stages now. What do you mean by that? This sporozoid injected into the body by the female Anopheles mosquito now undergoes several changes. As it is undergoing several changes, it will be causing the malaria. It will be causing the disease called as malaria. Now this sporozoid injected now into the body of a healthy person, and this sporozoid is carried by the blood. This sporozoid is carried by what? By the blood. Okay, by the blood to the liver cells and the RBC cells. Sporozoid is carried by the blood to the liver cells and RBC cells. Now, if you are looking at the sporozoid, please remember it will be appearing in sickle shape. This is also important. What is the shape, or how do you identify that it is a sporozoid? The shape of the sporozoid is sickle shape. Okay, the shape of the sporozoid is sickle shape. Remember that. Now, if you are looking at the sporozoid, okay, it will be like this. Okay, the sickle shape. Okay, it will be present as this sickle shape, and at the anterior end it has the apical cap. Okay, at the anterior end it has the apical cap. Okay, it is having the apical cap, and it has the cytoplasm. Okay, it is having the cytoplasm inside the sporozoid. Of course, it is a cell, and at the center it will be having what? The nucleus. Okay, nucleus. So let us say that this is the cytoplasm, and there will be cell organelles that are present, which I am not drawing here, and at the same time here paid, okay, tubes will be there. Paid tubes will be there. Paid tubes will be there that are opening into the apical cap. So this apical cap, where is it present? It is present at the anterior end. Okay, it is present at the anterior end. Please remember that. Apical cap is present at the anterior end, at which there are two tubules that are open, and please remember that this sporozoid is completely covered by an elastic. Okay, it is completely covered by an elastic microtubule. Please remember that the sporozoid is covered by what? The elastic microtubule. Okay, elastic microtubule. Okay, elastic microtubule. Okay, please remember this. Elastic microtubule is present around the sporozoid, which is in what shape? Sickle shape. Okay, sickle shape. And now here these tubules that are there here, okay, microtubules that are there, these tubules which are opening at the anterior end, okay, at the anterior end, called as the apical cap, So this is sporozoid. Okay, this sporozoid is showing two tubules. What are those tubules called as? Paid organelles. These are called as the paid organelles. Okay, these are called as paid organelles. And these paid organelles are otherwise, in simple, we can call these paid organelles as what? Secretory organelles. Okay, secretory organelles. And these secretory organelles will be producing cytolytic enzymes. They will be producing cytolytic enzymes. They will be producing cytolytic enzymes. What is the function of these cytolytic enzymes produced from the secretory organelles of what? The sporozoid. They help in penetrating the liver cells. So here you have to remember how these sporozoids are entering into the liver cells and RBC cells means it is because of the cytolytic enzymes. What do? Penetrates. Okay, penetrates the liver cells. Helps in penetrating the liver cells or RBC cells. Liver cells. Okay, penetrates the liver cells. Okay, penetrates the liver cells. So here this is the structure, a simple diagram showing this sporozoid. So here you have to remember the structure of the sporozoid. If your sporozoid is the infective stage of malaria, we are saying. And what is the shape? Sickle shape. And if you are taking this sporozoid, at the anterior end, it is having the apical cap. Into this cap, there are paid organelles that are opening. And these paid organelles are also can be called as what? Secretory organelles. And these secretory organelles are producing what? Cytolytic enzymes. And cytolytic enzymes will be, and these secretory organelles, okay, these secretory organelles, if you are seeing, will be producing what? Cytolytic enzymes. Cytolytic enzymes helps in penetrating the liver cells. That's how the sporozoid is entering into the liver cells. And undergoes several changes. And the same sporozoid, after undergoing many changes, enters into the RBC cells also, causing what disease? Malaria. That's the end point. Okay, we are trying to understand how this is completing its life cycle and causing malaria. So this is the simple structure. And please remember, this sporozoid is enclosed in what? Elastic microtubule. microtubule. Okay, elastic microtubule. Please remember this. Okay, elastic microtubule. So this is what we have to remember, and how the female Anopheles mosquito is introducing or inoculating the sporozoite into the human beings. Now we are going to see the different stages. Okay, we are going to discuss now different stages in the asexual cycle, or otherwise called as what? Schizogony in man. Okay, what are the different stages through which this sporozoite will undergo changes causing malaria? We are going to discuss as different stages. We are going to discuss. So here, asexual cycle or schizogony in man occurs in different stages. The first one, if you are seeing, okay, the first one, it is called as pre-erythrocytic. Okay, pre-erythrocytic schizogony. Okay, pre-erythrocytic schizogony it is called as. Okay, schizogony. And the second one is called as Exo-erythrocytic cycle. Okay, Exo-erythrocytic cycle. Okay, Exo-erythrocytic cycle. And the third one, what is the second one? Exo-erythrocytic cycle. And the third one is erythrocytic cycle. And the next one is post-erythrocytic cycle. Okay, post-erythrocytic cycle. And the next and the last one is formation of gametocytes. Okay, formation of gametocytes. Okay, formation of gametocytes. These are the different stages. Which we'll be studying under asexual cycle or schizogony in man. Okay, we'll pick up one by one to understand what are these pre-erythrocytic schizogony, exo-erythrocytic cycle or erythrocytic cycle, or post-erythrocytic cycle, or formation of gametocytes. We are going to discuss one by one. Now we will discuss about the first stage, pre-erythrocytic schizogony. Now in this, as soon as the infected female Anopheles mosquito bites a healthy person, it is inoculating or injecting what? Sporozoite. Okay, it is injecting sporozoite. This is what we said, infective stage of the malaria parasite. What is the shape of this? Sickle shape. So now the mosquito, what is that mosquito? The female Anopheles mosquito. Okay, female Anopheles mosquito injected what into the body? Sporozoite. Now the sporozoite is present in what? It is present in the blood. Okay, it enters into the blood now. Okay, sporozoites are present in the blood. These sporozoites will be present in the blood just for half an hour. Okay, just for half an hour they will be present inside the blood. Okay, only for half an hour they will be present in the blood, which means they will take just half an hour to reach to the liver cells. Okay, to reach to the liver cells, they just take half an hour. And let me be very clear, mosquito, when it bites, okay, what it is releasing? Sporozoites. And please remember, with one bite of a female Anopheles mosquito, it can release about 2000 sporozoites into the bloodstream of a human being. With one bite of a infected female Anopheles mosquito, it can release the 2000 sporozoites into the blood. And all these 2000 sporozoites will be taking just half an hour to reach the liver cells. So within half an hour, they will be disappearing from the blood now. Within half an hour, all these sporozoites will disappear from the blood. They are already reached what? The liver cells. Now here, they will be entering into the parenchymatous cells of the liver. They will be entering into the parenchymatous cells in the liver, present in the liver. So where are these sporozoites present in the liver? They are present in the parenchymatous cells of the liver. Now here interesting point that you have to make note is that why they will be immediately entering into the parenchymatous cells of the liver. It is because to escape the phagocytic cells called as leukocytes, to escape from the phagocytic cells called as leukocytes, they will be entering into the parenchymatous cells. Remember, why? The question is why the sporozoites will be entering into the parenchymatous cells of the liver. The answer is to escape from the phagocytic cells called as leukocytes. Because what is the function of leukocytes? If any pathogen enters inside into the body, they have to undergo phagocytosis, or they have to kill that particular pathogen by a process called as what? Phagocytosis. To escape that process, these sporozoites will be entering into the parenchymatous cells from the leukocytes. Okay, to escape from the leukocytes. Okay, leukocytes. Now, after reaching the liver cells, now they start multiplying. Now. So how they will be multiplying, we will be seeing now. So let us imagine. Okay, let us imagine that if this is the liver. Okay, if this is the liver. So inside the liver, we know that the liver is made up of what? Cells called as hepatic cells. We call them as. Okay, we call them as what? Hepatic cells. Hepatic cells. Liver is made up of what? The hepatic cells we see. Okay, hepatic cells. Now, how this sporocyte will enter into the hepatic cells? We already said at the apical cap of this sporocyte, it has two paired secretory organelles called as the producing cytolytic enzymes, by which it will be penetrating into the liver cells. It will be penetrating the liver cells. Now, after penetrating inside into the liver cells, the sporozoite, okay, the sporozoite, let us say that this sporozoite is present in what now? The liver. So this sporozoite now starts absorbing all the nourishment from the liver cells. Now once it absorbs this nourishment, it starts increasing in size. Now the same sporozoite, now let us call it as what? Trophozoite. Okay, trophozoite. Now sporozoite only called as what? Trophozoite. It is increasing in size. And the same thing, now trophozoite will be now increasing much more, and now it is called as schizon. It is called as what? The schizon. Now this schizon, which increased in size, okay, maybe let us say that this is the size, having the nucleus, schizon. Now this schizon is the mother cell, we call it as. It is called as what? Mother cell, which will be undergoing now asexual reproduction. This schizon, when it is undergoing asexual reproduction, what did we call it as? We are calling it as what? Schizogony. What do you mean by the word schizogony? It is a method of what? Asexual reproduction. What is that method of asexual reproduction taking place in the case of schizon? Multiple fission. It will be taking place. It will be undergoing multiple fission. Now this schizon, if this is the cell, this cell will undergo what process? Multiple fission. It will be undergoing multiple fission. See here, what do you mean by multiple fission now? Within one cell, the nucleus that is there, one nucleus, will divide into many nuclei. Okay, will divide into many nuclei. That we are calling it as what? Multiple fission. Okay, we are calling it as multiple fission. So multiple fission is what type of method? Asexual reproduction. Okay, it is a type of asexual reproduction. So this schizon, okay, the nucleus that is there present in this cell will undergo multiple fission and produce many nucleus. Okay, many nuclei will be produced. All these are what now? The nuclei. So each nuclei will be surrounded by what? The cytoplasm, and then this will get ruptured, releasing the nucleus having cells. Okay, these are all the cells which are produced now. Okay, which are produced, and the cells which are produced like this. Okay, like this by multiple fission. Okay, we are calling them as, next we will be calling them as what? The cryptozoids. We call them as the cryptozoites. Okay, we are calling them as cryptozoites, or otherwise we can also call them as cryptomerozoites. Okay, cryptomerozoites. Okay, cryptomerozoites. This is what we should remember when we talk about the pre-erythrocytic schizogony. Pre-erythrocytic schizogony. So here we are saying that cryptozoites, or otherwise called as what? Cryptomerozoites are the ones which are formed in pre-erythrocytic cycle or pre-erythrocytic schizogony. Pre-erythrocytic schizogony. So here, if you are seeing very clearly, each sporozoite, we said already 2000 sporozoites will be released by one bite of the mosquito. But each sporozoite will be undergoing these changes, changing into increasing in size, then we are calling it as trophozoite. And once it starts undergoing schizogony by a process called as schizogony means what? Asexual reproduction by a process called as multiple fission, producing what? Many merozoites. We can also call them as what? Merozoites. Please remember, we can call them as what? Merozoites. So how these merozoites are released? See, once the multiple fission is taking place, okay, each cell of the liver will be under pressure, okay, because of the multiple fission that is taking place. There will be pressure on the walls of every liver cell in which this schizont is present. This schizont is present because they are increasing in size and they apply pressure on the walls of the liver cells. And at the same time, what is happening? Multiple fission is taking place to produce what? The merozoites, called actually as what? The cryptomerozoites or cryptozoites. Cryptozoites or cryptomerozoites. So here, this process will be taking place, or it will be taking for about 8 to 10 days. Please remember this. Okay, pre-erythrocytic cycle, pre-erythrocytic schizogony will be taking 8 to 10 days to complete. Okay, to complete pre-erythrocytic schizogony, it takes 8 to 10 days. It takes 8 to 10 days. Now, in short, let us say what is pre-erythrocytic schizogony. What is pre-erythrocytic schizogony? It is the process in which single sporozoite, will be producing what? Cryptomerozoites. Okay, it is the process in which a single sporozoite will develop into what? Cryptozoites. Okay, will develop into cryptozoites. Where this process is taking place? In the liver. This complete process is taking place where? It is taking place in the liver. It is called as what? Pre-erythrocytic schizogony. Pre-erythrocytic schizogony. So here, the pre-erythrocytic schizogony means what? We already studied. You know now what do you mean by the word schizogony. Schizogony means what? Asexual reproduction that is taking place by what process? Multiple fission. Okay, multiple fission. So let us put it in words, in simple and short. What do you mean by pre-erythrocytic schizogony? Okay, the process of formation of many cryptozoites or cryptomerozoites. The process of formation of many cryptozoites from a single sporozoite in the liver cell is called pre-erythrocytic schizogony. Once again, the process of formation of many cryptozoites from a single sporozoite in liver cell is called pre-erythrocytic schizogony. So this is what is pre-erythrocytic schizogony. Now the next one is what? Exo-erythrocytic schizogony or exo-erythrocytic cycle. Exo-erythrocytic cycle. Now, exo-erythrocytic cycle means the cryptomerozoites that are released, from the liver cells will again go back and infect the healthy cells of the liver, fresh cells of the liver. The cryptomerozoites that are formed will now come outside. They burst out from that cell, liver cell, come out and now attack the fresh cells of the liver. And these cryptomerozoites will again undergo the same process and they again form now in the exo-erythrocytic cycle, which we are going to see now, will be forming metacryptozoites. cryptozoites. So here you have to remember pre-erythrocytic schizogony. Okay, pre-erythrocytic schizogony. The end structure that is formed is what? Cryptomerozoites. Cryptomerozoites. Now we will discuss about the next one called as the exo-erythrocytic schizogony. Exo-erythrocytic schizogony, or otherwise exo-erythrocytic cycle. Exo-erythrocytic cycle. So what is happening here now? In the first one we studied that what are formed? The cryptomerozoites. Okay, cryptomerozoites. These cryptomerozoites will attack the fresh liver cells, and form as what? Now see here, the cryptomerozoites attacking the fresh liver cells, and they will be forming as what now? The schizon. Okay, they are repeating the same cycle. Okay, they form as the schizon and develop into now metacryptozoites, Okay, this is the second cycle. They completed first generation and now the second generation, metacryptozoites. And these metacryptozoites that are formed, okay, maybe of some metacryptozoites may be small, okay, some metacryptozoites will be small and some will be large in size, okay, some are large in size. What are small and what are large? Metacryptozoites. Those metacryptozoids that are small will be called as what? Micro metacryptozoids. Okay, micro metacryptozoids they are called as. And those which are large, okay, those metacryptozoids which are large will be called as macro metacryptozoids. Okay, cryptozoids. These are called as macro metacryptozoids. Now there are two based upon this science, metacryptozoides that are formed. Okay, that are formed now. So here the micro metacryptozoids, okay, that which are small, okay, as the word indicates, micro means what? Small. Those micro metacryptomerozoids or cryptozoids will now go and attack the RBC cells. They will go and attack the RBC cells. Please remember that. Micro metacryptozoids which are small in size will go and attack the RBCs. Then what is the role of the macro metacryptozoids which are large in size? Again these, okay, which are large, again they attack what? New liver cells, okay, fresh liver cells, again they are attacked by the macro metacryptozoids to repeat the same cycle to produce what? The micro metacryptozoids. Otherwise in simple we are calling them as what? Merozoids, okay, don't get confused. Merozoids we call them as. So here there are two that are formed, okay, and this is what is taking place in exo-erythrocytic schizogony or exo-erythrocytic cycle. So in simple words, to put it in simple words, we can say that the process of formation of metacryptozoids, okay, the process of formation of many metacryptozoids from single cryptomerozoid in the liver cell is called as what? Exo-erythrocytic schizogony. So here, what do you mean by exo-erythrocytic schizogony? When many metacryptozoids, okay, are formed from what? Cryptomerozoids. It is called as what? Exo-erythrocytic schizogony. Okay, exo-erythrocytic schizogony. And please remember, you have to make note of this, that exo-erythrocytic schizogony will not be seen in the malaria which is caused by Plasmodium falciparum. So we have to make note, okay, that the exo-erythrocytic schizogony, is not seen, is not seen in Plasmodium falciparum, Plasmodium falciparum. Please remember this, falciparum. This is This exo-erythrocytic schizogony will not be there present in what? Plasmodium falciparum. Plasmodium falciparum. So these are the points that we have to keep in mind when we are talking about exo-erythrocytic schizogony. Exo-erythrocytic schizogony. So study. And please note, how many days this process will be taking place? Exo-erythrocytic cycle will take how many days? Please remember, it will take only 2 days to complete. Okay, it will take only 2 days. Okay, 2 days to complete the process of exo-erythrocytic cycle. Okay, exo-erythrocytic cycle. It takes only how many days? Only 2 days. Okay, only 2 days. So now the next one is erythrocytic cycle. Erythrocytic cycle. So what do you mean by erythrocytic cycle? It is the process of formation of merozoites. Process of formation of merozoites from a single micrometa-cryptozoid. From a single micrometa-cryptozoid is called as erythrocytic cycle. So here we have to remember that merozoites are formed from a single micrometa-cryptozoid. That is called as what? Erythrocytic cycle. Now, erythrocytic cycle will be taking place in the RBC cells. Where this process is taking place? It is taking place in RBCs, okay, red blood corpuscles. And please remember, single micrometa-cryptozoid will attack single RBC cell. Single RBC cell. So you should make note that all micrometa-cryptozoids will not attack only one RBC cell. One micrometa-cryptozoid will attack one RBC cell. So here we are seeing, let us say that this is the micrometa-cryptozoid and this is what? The RBC cell. Okay, RBC cell. So one single micrometa-cryptozoid attacks the RBC cell. Once it attacks the RBC cell, it enters inside and now it is changing into oval in shape, okay, spherical in shape. Now the parasite, okay, the micrometa-cryptozoid is in spherical in shape. Once it is there in spherical in shape, now this stage we are calling it as what? Trophozoid. We are calling it as trophozoid. Now this micrometa-cryptozoid starts absorbing the nourishment from the RBC cells. Okay, from the RBC cell. What is the nourishment present in the RBC cell? It is nothing but the hemoglobin. Okay, hemoglobin that is present. So now what this does is that it will digest the hemoglobin into what? It will digest the hemoglobin into heme. Okay, into heme and globin. Globin is the protein part. So this parasite micrometa-cryptozoid will absorb the globin. Okay, it will absorb the globin. And now once it is absorbing the globin, slowly there will be formation of a vacuole present in the RBC. Okay, there will be formation of a vacuole that will be formed. Okay, there will be formation of a vacuole that will be formed and pushing the nucleus towards the periphery. Pushing the nucleus towards the periphery. Pushing the nucleus to the periphery. So here is the nucleus. Okay, here is the nucleus. So once the vacuole is formed, now here what happened? It is appearing like a signet ring stage. It is appearing like a signet ring stage. And now slowly this signet ring stage now slowly develops into a amoeboid stage. Now here is the parasite. Okay, parasite. Now it is in what shape? Amoeboid stage. This amoeboid stage now slowly engulfs so much of this hemoglobin, digests the hemoglobin and takes it as the nourishment. Okay, digests the hemoglobin and takes it as the nourishment and increases in size. It will increase in size. So here we have to remember the amoeboid stage. Okay, amoeboid stage. So this amoeboid stage, as the parasite micrometa-cryptozoid, as it is undergoing many changes and increasing in size, now we are calling it as what? Amoeboid stage. It digests so much of this hemoglobin and increases. And because this parasite is increasing, even the size of the RBC also will increase. Even the size of the RBC also will increase. So now increase in the size of RBC due to the amoeboid stage of the parasite can be said as what? It can be called as what? Hypertrophy. We call it as what? Hypertrophy. Hypertrophy means increase in size of the RBCs. Increase in size of the RBCs is called as hypertrophy. Why there is increase in size of the RBC? It's because of the amoeboid stage of the parasite. Amoeboid stage of the parasite. And slowly from the amoeboid stage, it now reaches to another stage called as what? This schizon. It is completely enlarged in size now. This schizon, okay, the parasite. Now this schizon, now the word itself indicates that now schizon will undergo what process? Schizogony means what? Asexual reproduction. It will undergo asexual reproduction now. So now this schizon of the parasite will undergo asexual reproduction by multiple fission and produces many parasites called as now merozoites. Now merozoites are formed. Okay, merozoites are formed. So there are many merozoites that are formed by schizogony. So what is the process that is taking place here? Schizogony will be taking place. So by a process called as schizogony, there will be many cells that are formed from single micrometacriptozoid. That many cells are called as what? Merozoites. Merozoites are formed. And all these merozoites are present towards the periphery of the RBC cell. When they are present at the periphery of the RBC cell, now they are appearing like a rose flower, the petals of a rose, okay, which are there like a ring, okay. They are just like petals of a rose that will be there like a ring that are seen in a rose flower. They will be present. So that's why we call this arrangement as what? Rossetti ring stage. Okay, we call this arrangement as what? Rossetti ring stage. Okay, Rossetti ring stage we call it as. Okay, Rossetti ring stage. So why it is called as Rossetti ring stage? It's because the merozoites that are formed by a process called as what? Schizogony means multiple fission. They are appearing as the petals of a rose flower. So it is called as what? Rossetti ring stage. And now these merozoites will be applying pressure on the walls of the RBC and they will rupture the walls of the RBC. And once they rupture the walls of the RBC, now these merozoites will be released. Okay, these merozoites are released, and again these merozoites will attack again the fresh RBCs. The fresh RBCs and continue the same cycle in increasing the number of merozoites. And here the heme that is there is toxic. Okay, soluble form of the heme is toxic, and this malaria parasite will be converting this heme into hemozoin. Okay, into a hemozoin substance. Okay, it is a malarial pigment we say hemozoin. Okay, from the heme, the parasite is converting the heme into hemozoin, malarial pigment we say. And please note that this hemozoin, okay, in various studies they say that hemozoin protects the malarial parasite from the immune system of the body. Hemozoin protects the merozoites or malarial parasite from the leukozoites or those cells which will destroy this parasite. Okay, it is the parasite. The parasite is protecting itself. The parasite is protecting itself from the immune system by producing what? Hemozoin. So here we have to remember the process of formation of merozoites from micrometacriptozoid is called as what? Erythrocytic cycle. And this process will be taking 48 hours. Okay, it will be taking 48 hours to complete. Okay, it will be taking 48 hours to complete this cycle called as erythrocytic cycle. Okay, erythrocytic cycle. Now the next one is post-erythrocytic cycle. So in this, it is very simple. This cycle is very simple. We studied that now merozoites are formed. Okay, merozoites are formed. They are formed in what? In the previous one, what we said in RBCs. Okay, in RBCs, what are formed? Merozoites. So here, what we have to remember is that these merozoites that are produced in the RBCs, okay, produced in the RBCs, these merozoites will now migrate, okay, they migrate back to the liver, okay, they migrate back to the liver cells, and they undergo what process? Here in the liver cells, they will be undergoing schizonic process, okay, schizonic process or schizogony. They will be undergoing schizogony to produce many number of these cells, okay, called same cycle it will be repeated. So here you have to remember that under the post-erythrocytic cycle, the merozoites that are produced in the erythrocytic cycle will again migrate to the liver cells and undergo what? Schizogony to produce the same type of cells. This process will be taking place. So this is called as what? Post-erythrocytic cycle. Now the next one is formation of gametocytes. So what we discussed in the previous one, especially about the erythrocytic cycle. In the erythrocytic cycle, okay, the merozoites, are formed. Okay, merozoites are formed in what? Are formed in RBCs. Okay, in RBCs we said. Okay, this is in the erythrocytic cycle. And later what we said, some merozoites will again go back to the liver we said. And some of the merozoites now they are of different size. Some merozoites will be called as will be changing. Okay, some merozoites will be changing into microgametocytes. Okay, microgametocytes. Okay, merozoites are changing into microgametocytes and also into macrogametocytes. Okay, micro and macrogametocytes. These microgametocytes and macrogametocytes are the ones which will be developing into what? Male and female gametes. Microgametocytes will develop into the male gametes. Okay, whereas the macrogametocytes will be developing into female gametes. And this whole process will be taking place within the RBCs. Okay, formation of microgametocytes and macrogametocytes. This whole process is also taking place in what? In RBCs. But here we have to remember in RBCs which are present in the spleen, okay, which are present in the spleen or bone marrow, So here please remember that the RBCs which are present in the spleen or bone marrow, in those RBCs what are formed? The merozoites are converted into microgametocytes which will be changing into male gametes, and merozoites are changing into macrogametocytes which are changing into female gametes. And please remember this. Okay, and these two are very important. Okay, these two are very important, and please remember that these two are formed in the RBCs which are present in the spleen and bone marrow. And these gametocytes, will die if they are not transferred to the mosquito within a week. Please make note of that. These gametocytes that are formed in the human beings, in the RBCs, if they are not carried or transferred into the mosquito within a week, they will die. They will get destroyed. So the male gametes and female gametes will be there surviving in the blood only for 1 week, within a week, and within that week they have to be picked up by what? The mosquito. Okay, so mosquito will be taking what? The male gametocytes and female gametocytes into its body. There the fertilization will be taking place. So till here, this is called as what? Asexual cycle or schizogony that is taking place in the man. Okay, So what is formed at the end of asexual cycle? The male gametocytes and female gametocytes. Okay, male gametocytes and female gametocytes in the RBCs present in the spleen and bone marrow. And these male gametocytes and female gametocytes have to be carried to the mosquito within a week. Okay, within a week. Otherwise, they will die. Thank you so much.